Now we look at carbohydrates. These are broken down to CO2 in a process called oxidation. These are known as combustion and can release large amounts of energy. The principal source, and this is a principal source for cellular metabolism. So what is a monosaccharide? This is a simple sugar that is a single carbohydrate molecule. Important monosaccharides are going to be fructose, glucose, and ribose. And two monosaccharides that are bonded together, these are known as disaccharides, and some can form oligosaccharides, and many form polysaccharides. So now we'll look at glycosidic linkage. So glycosidic linkage is the bond between two sugar molecules. These are formed covalently in a dehydration reaction that requires enzymatic catalysts. So when we attach sucrose and fructose, we're going to get glucose. Anomeric carbon is pointing down. So if we draw this right here. Let's redraw this. So as we can see, it's pointing down, which means there's an alpha 1-2 linkage. So I apologize. On the left we have glucose, and then we have fructose on the other side, and this is going to form sucrose. So the anomeric carbon is pointing down, which means alpha 1-2 linkage, glucose, alpha 1-2 to fructose. And these are going to be disaccharides. So how are glycosidic linkages named? According to which carbon the sugar molecule comprises the linkage. So alpha or beta is also specified. So here we have lactose, which is milk sugar. This is a disaccharide joined in a galactose, beta 1,4 glucose linkage. Lactose has this beta linkage, and polymers made from disaccharides form important biological macromolecules. An example is glycogen. This serves as an energy storage carbohydrate in animals. This is composed of thousands of glucose units. They are joined in alpha 1,4 linkages, and alpha 1,6 branches are also present. Starch, this is like glycogen. The branches are different. This serves some purpose in plants. Cellulose is a polymer of cellobiose, but cellobiose is not found freely in nature. So this exists only in polymerized form. Beta-glycosidic linkages allow polymer to assume a long, straight, fibrous shape, and wood and cotton are made of cellulose. So now we'll look at the hydrolysis of glycosidic linkages. Hydrolysis of polysaccharides turns into monosaccharides. These are favored thermodynamically. Hydrolysis is essential in order for sugars to enter metabolic pathways such as glycolysis and be used for energy by the cell. There must be an enzymatic catalyst, or hydrolysis doesn't occur at a significant rate. Different enzymes catalyze hydrolysis of different links, just like peptide bonds and amino acids, such as like protease. But this is for carbohydrates. So these enzymes are named for the sugar they hydrolyze. An example for maltose is maltase. And maltase is the enzyme that catalyzes the hydrolysis of maltose into two monosaccharides. So the fact that these enzymes are specific make it a great example for the significance of stereochemistry. Example, for cellulose, a cotton t-shirt is pure sugar. However, we can't digest it because our enzymes don't break down the beta glycosidic linkages found in the cellulose. So cellulose is the energy source found in grass and hay. And even though cows are mammals who lack enzymes to break down cellulose, cows have an extra stomach called a rumen to digest cellulose for them, as humans can't because they don't have this extra stomach and extra rumen to help digest cellulose. But there is a carbohydrate called lactose, and humans can actually digest lactose, which is a beta linkage, and we carry a specific enzyme that digests lactose, and this is called lactase. But this is the exception to the rule because mammalian enzymes don't hydrolyze beta linkages. We only hydrolyze alpha linkages. So lactose malabsorbers, these are going to be people that don't have the enzyme lactase. Therefore, the lactase enzyme, lactose they consume ends up in the colon, creating gas or diarrhea, which means people are lactose intolerant. So when we're children, we actually make lactase as children so we can digest our mother's milk. But as we get older, we can naturally stop making this enzyme and become lactose intolerant, which requires net energy input. This is going to be polysaccharide synthesis or hydrolysis. So let me restate this. Which requires net energy input, polysaccharide synthesis or hydrolysis. Hydrolysis is thermodynamically favored, therefore energy input is required to drive the reaction toward polysaccharide synthesis. If the activation energy of polysaccharide hydrolysis were so low that no enzyme was required for the reaction to occur, will this make polysaccharides better energy storage? No, because polysaccharides would hydrolyze spontaneously. There's high activation energy of polysaccharide hydrolysis, which allows us to use enzyme as gatekeepers. When we need energy from glucose, we open the gate of the glycogen hydrolysis.